Everyone, and welcome back. This is part 4 of our series on the CGIAR Root Volume Estimation Challenge. If you've been following along, you know we've already covered quite a bit. We visualized segmentation masks, improved a CNN benchmark model, and even tackled YOLO for segmentation. But this video is special. We're going to address a major pain point and unlock a crucial insight that will change how we look at the data, literally. Up until now, one of the biggest challenges has been visualizing the individual plants. Remember, each image can contain up to 7 cassava plants, scanned from both the left and right sides with ground-penetrating radar. It's been really hard to visually separate those 7 plants and get a clear picture of what's going on. Yesterday, in the Q&A video posted by the Zindi team, we hit upon a key insight. To truly see the complete plant structure, we need to take the right side image, rotate it 180 degrees counterclockwise, and place it above the left side image. This notebook is all about implementing that insight and finally getting a clear view of our plants. Okay, let's get started. First up, let's look at the functions defined in this notebook. We have draw vertical lines. What this does is, it takes an image and draws vertical lines across it. This effectively divides the image into sections. It also helpfully labels each of these sections with a number, starting from 1. This is going to be useful for us because, remember, each scan can potentially show up to 7 different plants. This function helps us visually separate them. Next, and this is a really important one, we have merge left-right. Okay, so pay close attention here. Remember from the problem description, the radar scans the cassava plants from both the left and the right sides. This function, merge left-right, takes a left side image and a right side image as input. It then merges them into a single, combined image. It does a couple of clever things. First, it crops both images to make sure they have the same width. Then, and this is the key part, it rotates that right side image 180 degrees counterclockwise and places it on top of the left side image. This is exactly the insight we had from the Q&A. It's how we get the full picture of the plants. After that, we've got stack images vertically. This one's pretty straightforward. It takes a list of images, and it stacks them one on top of the other, vertically. You can think of it like creating a tall panorama or a collage where each image is placed below the previous one. This will be helpful for us to see how the plants change at different depths. And finally, we have See All Plants. This is our main function for visualizing everything for a single folder. It does a few things. First, it finds all the left side and right side scan images within a given folder. Then, it takes each corresponding left and right pair, and it uses our merge left-right function to combine them. Finally, it takes all those merged images and uses stack images vertically to stack them all up, giving us a complete view of that folder's scans. Moving on, we load our training data. This is the train data file, and it contains all sorts of important metadata about our images, things like the folder names, the plant number, which side was scanned, left or right, the starting depth of the scan, and the ending depth. This metadata is crucial for organizing and understanding our data. Then, we do a little bit of data exploration. We're using value counts to see which folders have the most entries in our training dataset. This is just a quick way to get a sense of which folders might have the most complete scans or perhaps represent more common experimental setups. Okay, now we're getting to the exciting part, actually visualizing the data. This is where the magic happens. First, we pick a folder to visualize where the pattern is most obvious. Then we call our See All Plants function, passing in the folder name. This function, as we discussed, grabs all the relevant images, merges the left and right pairs, and stacks them vertically. Next, we use draw vertical lines to draw those vertical lines on our combined image, separating the potential regions where each of the 7 plants might be. And then, boom, we display the image. This is the moment of truth where we finally get to see the data in a meaningful way. You can clearly see the 7 plants. As you look from top to bottom, you're essentially looking deeper and deeper into the ground, and you'll be able to see how the structure of the plants change with depth. Finally, we have this code block that's set up to loop through all the folders in our training data. For each folder, It'll create the visualization we just talked about, display it, and also show you the corresponding rows from the training data. It's a great way to quickly inspect your data and get a feel for the different root structures. The code also prints out the folder name and the range of depths that were scanned for that folder, giving you even more context. We can see that patterns are not always obvious. There is still a lot to do in this challenge. And that's it for this part. We've finally cracked the code on visualizing the ground-penetrating radar data in a way that reveals the full plants. We took that crucial insight, rotating and stacking the right-side images, and built functions to make it happen. Merge left-right is the star of the show here. Now we can clearly distinguish the individual plants and see how they change with depth. This visualization isn't just pretty pictures. It's essential for understanding the data, validating our models, and even generating new ideas for feature engineering. We've overcome a major hurdle that was holding us back in the previous videos. In the next part, we'll leverage this new understanding to build even better models. Perhaps we'll revisit our CNN or YOLO approaches with this clearer view of the data. The possibilities are exciting. Thanks for watching, and I'll see you in the next video.